Hi, sixth graders. How's it going? This is Mr. Miles coming at you with the podcast as they make their triumphant return back to prime time. Now, here we go. So, what we're going to talk about today is we're going to talk about movement with a cell. And when we talk about movement, we're talking about this outside part, which is the cell membrane. And this is a cell membrane, and we're going to talk about how things get in and how things get out of the cell membrane. Remember, we said that this is selectively permeable, so certain things can come in, certain things can go out, but other things can't. Some things can only travel in one direction, some things travel in two directions, some things can't travel in any directions. Here we go. So let's talk about it. Now, the thing to understand is there's going to be two types that we're going to be talking about. There's going to be the type that uses energy and the type that doesn't use energy. Now, there's two types that will not use the energy of the cell, and we're going to be showing those in a second. But this thing decided to stop working. There it goes. Here we go. So, the two types we're going to deal with is we're going to deal with passive transport and then active transport. So remember, active transport, energy, passive transport, no energy. And passive transport splits off into another one, so you can have passive transport or you can have facilitated diffusion. And how these ones work is, here you have your cell membrane, and you have ones like H2O. These are very small molecules, so they can actually move freely back and forth through the cell. And they're going to follow the diffusion flow, so if there's a lot more H2O on this side, then they're going to be able to flow to that side. And if it's vice versa, it's going to flow the opposite way. Remember, it goes from the area of high concentration to an area of low concentration. Facilitated diffusion works in the same way, area of high concentration, low concentration, but it deals not with H2O, but larger molecules. Like let's say maybe we have sugar, and it needs to get the high concentration sugar into the low concentration cell. Well, they can't just freely go across there because they're too big. So instead, you have protein channels. So there's a protein channel. And it's like an opening in the cell membrane that's specially designed so only sugar can fit through it. Makes perfect sense, I'm sure. Now, you can come across other molecules that are even bigger than that, and they are not able to actually, well, not that they're bigger than that, they're not able to go through the cell membrane because of this particular problem. On the inside of the cell, you have a high concentration, and on the outside, you have a low concentration, so let's say you only have that much. And so naturally, it's going to want to flow out of the cell. Well, there's a way to work against that. You can take another one of these protein channels, and using energy, you can actually use energy to pull that inside. Now, when you get to high school and later, they're going to go more over what exactly is happening and what kind of energy is being used in order to pull that over. But what it does is it goes against what normal diffusion would say, because it would normally, again, want to go this way. Well, it goes from the area of low concentration to an area of high concentration as long as you're using energy. All right, remember that the example we gave was like one of riding up a hill. Passive transport is kind of like riding down the hill. You don't have to use any energy because here's the area of high concentration, go to an area of low concentration. Well, if you want to go up the hill, an area of low concentration, an area of high concentration, you actually have to use energy, and that's using your legs, which causes you to sweat and pant. It's really terrible. Now, I accidentally pressed that again. I didn't really want to because it takes forever. Well, the last two things we're going to talk about is we're going to talk about endocytosis and exocytosis. I'll give you a little preview while we wait for this to load. Now, endocytosis, remember the word endo. And endo, simply what it means is inside. And so when we're talking about endocytosis, we're talking about bringing something inside. And exocytosis, remember, exo means to exit. So we're talking about getting rid of something. Well, you come across things. And now it's possessed by the devil. Because it does not do what I'm asking it to do. Oh, this thing hates me. When you're dealing with endo or exocytosis, you're dealing with particles that are too big to naturally actually go into a cell. So what the cell will actually do is it will envelop the particle that it wants to take in, or it will just push out any particle that's too large and it wants to kick out. And that's pretty much how endo and exocytosis is. I don't think I'm going to get this thing working, so hopefully that definition gives you enough. It's just large particles. And remember, it's not going to be like the size of sugar molecules. It's much larger than that. And that's why it has to perform those different types of cytosis. thing doing what I want it to do. All right, so that's the first podcast. If you have any questions, make sure you email me.